participant, we are starting in a moment. We are waiting for another speakers to join. We have Dr. Chabria in the room, so like we are waiting for Dr. Savle. So we have a very exciting announcement for you. So that will be revealed by Dr. Savle. So we'll wait.
Dr. Nipa.
I think he joined.
Dr. Nipa છે. નિધિ અને પાર્થનો ચોથો અંશ એટલે Nipa.
Hello. Ma'am, can you close your audio, please? Can you off the video, please? Sorry, audio, please.
Yes, we called over both of those things behind you.
I think now we are in.
Hi, Vivek.
Yeah. Hi, everyone. Sorry for the interruption. It's due to some technical glitches. Dr. Savla is now supposed to join, but facing some issues. So now we resolved it. So I hand over to Dr. Savle for an exciting announcement for you. It's over to you, Dr. Savle.
So Saule sir just left.
Okay, then we need to wait for a moment. Ma'am, we can start with next session. I think we'll take the announcement at the end of this session. Because
Yes, sir.
Yeah. So, you can start, ma'am. I request Dr. Pinaki to start his audio and video, so students can see him, and request Dr. Divya to take the further proceedings. So it's my privilege to introduce Dr. Pinaki Basu. We both work together at Lupin. So currently, Dr. Pinaki is working as a senior research scientist for the biotech formulations at Lupin Limited, and he has a vast experience for biophysical characterization of proteins and maps. And I hand over now to Dr. Pinaki to introduce Dr. Carpenter. Thank you very much, sir, for joining in a very short notice. We'll be delighted to have your thoughts towards the end of the session for Dr. Carpenter's talk. Thank you. Sir.
Thank you, Vivek. Am I audible?
You are completely audible.
Okay, thank you, Vivek. I appreciate the kind introduction. It's a very good evening to everybody. I would like to introduce Dr. Carpenter for the next presentation. Professor John F. Carpenter is a professor of pharmaceutical sciences at the University of Colorado School of Pharmacy and a co-founder and co-director of the University of Colorado Center for Pharmaceutical Biotechnology. His research interests include mechanism for protein degradation and stabilization in pharmaceutical formulations, during bioprocessing, and in drug delivery systems. Also, he has worked for several years to define rational strategies for stabilizing proteins and vaccines during freeze drying and storage in the dried solid. He has published more than 300 peer-reviewed papers and is an inventor on more than 30 issued patents. He is editor for reviews and commentaries for Journal of Pharmaceutical Sciences. He serves on the editorial advisory boards for pharmaceutical research, the AAPS Journal, Journal of Pharmaceutical Sciences, Current Pharmaceutical Biotechnology, Molecular Pharmaceutics, and Biopharm International. He has received several teaching awards and the Ebert Prize. He is a fellow of the American Association for Advancement of Science, the American Association for Pharmaceutical Scientist, or APS, and the National Academy of Inventors, and has received the AAPS Research Achievement Award in Biotechnology. He also is the organizer for the Colorado Protein Stability Conference, which is a well sought after conference. So with that, I would like to get the next presentation started for Dr. Carpenter.
Hi, this is John Carpenter. I'd like to thank the organizers for making this presentation for the conference. I'm going to focus today on fundamental mechanisms of protein aggregation and stabilization during freezing, drying, and solution and at interfaces, and I also will provide some practical examples of why it's valuable to know these mechanisms. These are many of my collaborators I've worked with over the years on some of these more mechanistic studies. And here are the topics we're going to cover today. But let's start with the practical example. These are two case studies. These are real studies from industry. In the first case, monoclonal antibody bulk drug substance was thawed. It was very cloudy upon thawing due to protein precipitation. If the precipitate was collected by centrifugation, IR spectra could be taken, which showed the protein molecules in the precipitates were completely native. If the thawed solution uncentrifuged was allowed to stand for a few hours at room temperature, the precipitates dissolved and the protein molecules went back into solution. The second case is quite a bit different in the end. The solution was cloudy upon thawing, and if it was let to sit out at room temperature for a few hours, the protein molecules did not go back into solution. And if the precipitate was collected by centrifugation and examined with infrared spectroscopy, the spectra indicated non-native protein molecules as well as a high content of intermolecular beta sheet, indicating the protein molecules had irreversibly aggregated. So reversible precipitation in case 1, irreversible precipitation and aggregation in case 2. So for these two cases, in which would the inclusion of 0.3 molar sucrose reduce the amount of precipitation? That is, in which case would the addition of this sugar be an effective formulation strategy? And here are the answers that you can choose from. We will go back over this later after we go through some of the mechanisms that are most relevant. But please note answer E, which is seemed to be very common in the industries. We don't really know the answer till we hire a bunch of consultants and do further extensive root cause analysis. However, I hope to convince you that by knowing mechanisms, you can say right away in which case sucrose would be a good formulation strategy. So let's start by talking with protein stability in solution and about how strongly binding ligands affect thermodynamic stability. This is called the Wyman linkage function that says if a ligand binds more to one state than another, that state with the bound ligand will be favored. In terms of protein stability, you can see here if we have the native protein going to the denatured state, if we add a ligand to the native protein that binds very strongly, the barrier between the native protein and the denatured state goes up a lot because ligand binding to a protein state lowers the free energy. By lowering the free energy, you favor that state, but in our example here, by lowering the free energy of the native state and not the denatured state, we get a much larger barrier between native and denatured state. And this is called the Wyman linkage function. In terms of practical use of this mechanism, here's an example with heparin, stabilizing fibroblast growth factor. Heparin binds very strongly to the native state, but not to the denatured state. And depending on the weight ratio, you can see a large increase in the apparent melting temperature for this protein. But there are many stabilizers that are not acting by binding strongly as ligands to the native state. These are what we think of as non-specific stabilizers like sucrose. These compounds essentially stabilize everything, all proteins. And the person that figured this out was Professor Serge Timoshev, our late friend and colleague. who worked for many years to understand the interactions between non-specific stabilizing additives like sucrose and proteins. And this is essentially the mechanism he defined. It's called the preferential exclusion mechanism. So in this case, if we have a stabilizer such as sucrose with a protein, and we do some studies, we find that the protein has a hydration shell, but there's a deficiency of the stabilizer around the protein compared to the outside. So in thermodynamic terms, this is a negative binding. So the negative binding means in the presence of the stabilizer, as shown here, the protein chemical potential increases. So preferentially excluded solute with the native protein increases the chemical potential of the protein. If that's all that happened, this would be a destabilizer. But the degree of exclusion depends on the surface area of the protein exposed to the solvent. So at the unfolded protein, we have a much larger level of preferential exclusion, which means a much larger increase in chemical potential. So with the native state, excluded solute leads to a little increase in chemical potential, whereas with the denatured state, we get a much larger increase. The net result is a large increase in the free energy of unfolding. Another way to look at this is from the principle of Le Chatelier that says if something perturbs the thermodynamics of a system, the system will tend to minimize that. So in this case, preferential exclusion increases protein chemical potential. The system will favor the native folded protein because the surface area of exposure to the solvent and the degree of exclusion is much less for the native than the denatured protein. In practical terms, we see this as an increase in free energy of unfolding. These are data for granulocyte colony stimulating factor. With guanidine unfolding, as you increase sucrose concentration, you increase the amount of denaturant needed to.
Sorry for the interruption, but one of the speakers' mic is on in the speaker room, so I kindly request you to please mute yourself.
To unfold the protein. Similarly with thermal unfolding, with heating, the more sucrose you have, the higher temperature you have to go to the unfolded protein. And this applies to inhibition of aggregation. When proteins aggregate, they have to go through a partially unfolded state, referred to as N star here, which then, if it interacts with the neighbor, potentially can form clumps that make aggregates. If you do this in the presence of a preferential excluded solute, the surface area for N star is greater than N, so the net barrier between these two states increase. So the activation energy for aggregation will go up in the presence of compounds like sucrose. And again, practically, we can look here, this is GCSF loss of monomer during days of incubation at 37 degrees at pH 7. And if you have higher and higher amounts of sucrose in the solution, there's less and less aggregation. An indication of the effect of sucrose on the level of N star is shown here. N star will have hydrogen deuterium exchange occurring at a faster rate than the fully compact native state because N star has more solvent exposure. And looking at H to D exchange, in the presence of zero molar sucrose, you get this amount of change in an infrared spectrum signal that's indicative of exchange. With more and more sucrose, you get less and less exchange because sucrose is reducing the level of this aggregation component N star. And this is all summarized here. The native state going to a partially unfolded state, this structural change is a key component of aggregation. But there's a second governing factor, and that's the intermolecular energetics, which are often indicated by a measured parameter called osmotic second virial coefficient. The main component playing a role here is charge-charge repulsion. Therefore, pH is a critical parameter governing protein aggregation. And here's an example of the importance of the so-called colloidal stability. Again, looking at the protein monomer loss as a function of time at pH 7. In this experiment, you can see it here. Sucrose slightly inhibited that. For this experiment, the important observation was benzyl alcohol greatly accelerated aggregation loss of monomer at pH 7. The reason for this is that benzyl alcohol causes a partial unfolding of the protein, which then fosters a high level of aggregation. Interestingly, well, first off, this can be partially blocked by sucrose, which inhibits some of that unfolding. But at pH 3, in the presence or absence of benzyl alcohol, the protein doesn't aggregate at all. That's because the protein's highly charged at pH 3, has its native structure, but the high charge keeps the molecules from interacting very well. Benzyl alcohol will perturb protein structure at pH 3, but the high charge inhibits aggregation. I wanted to have one little aside here about heating studies. Just keep in mind, when you have a heating study, you get both unfolding and aggregation for most of the molecules we work on. So either the free energy of unfolding, the native state stability, and our interactions between molecules can be the governing effect. So if you're doing a study, like say across pH, you may find the impact of pH at both steps, and that can help you overall find the optimal pH for a protein. If we put sucrose in the formulation, it most likely will influence this step by increasing the native state stability of the protein. Excuse me. So during freezing, we basically are forming ice, and the protein is in the non-ice phase. And protein aggregation will occur because proteins are perturbed by the various stresses that occur during freezing. Also, the high salt will favor intermolecular interactions. And so the degree of unfolding during freezing can be impacted by the native state stability as well as colloidal stability, or the aggregation. And any unfolding that occurs during freezing obviously will contribute during freeze drying. So how does the protein get stabilized by additives during freezing? Well, during freezing, the protein goes to the non-ice phase with water, unfrozen water. So essentially, everything's still the same as it would be in unfrozen aqueous solution, and a preferential exclusion mechanism applies. And so the degree of protection during freezing depends on the initial bulk concentration of the additive, just like it does with excipients in non-frozen aqueous solution. Here's some practical advice for freezing. Choose your buffer carefully. You want to make sure the buffer you choose does not have a large freezing-induced pH shift. And the buffer that's most notorious for these pH shifts is sodium phosphate. During freezing, the dibasic salt of the buffer can crystallize, and that crystallization can leave you with pure monobasic sodium phosphate and a pH down to pH 4 or so. The magnitude of the crystallization depends on the solution conditions, the temperature, and all the handling. So in general, using sodium phosphate, it should be avoided because you can get some surprises sometimes. For example, shipping of a frozen solution can cause buffer salt crystallization that you didn't see without shipping. During freezing, to maintain stability, having the appropriate pH is important, and also if you have stabilizing ligands, that's great. Non-specific stabilizers are often needed, like sucrose or trehalose, and it's been shown in numerous studies that non-ionic surfactants are also important to reduce aggregation, in this case most likely by reducing protein interactions with the ice-water interface. What about during drying? Well, now we have a completely different situation. As we dry a protein, the water molecules that hydrogen bond to the surface of the protein are removed. And this fosters protein unfolding. It may be also causing new intermolecular and intramolecular bonds to occur. And when you have unfolded proteins in a dried powder and you add water, there's a competition between molecules for refolding and aggregation. Also importantly, if you have unfolded protein molecules in a freeze-dried formulation, the storage stability of that product can be compromised. So how does additives like sucrose and trehalose protect during drying? Well, during drying, you literally strip off the hydration shell of the protein. At the water contents we have for a lot of lyophilized protein products, there's no water left around the protein. So we're not trapping water around the protein with the sugar. Instead, as the water molecules are removed, molecules like sucrose will hydrogen bond to the protein in place of the lost water. Now, for this to occur, the sugar has to remain amorphous. And the amorphous sugar is important because that allows the sugar to interact with the protein. If the sugar crystallizes, that can occur. But just having an amorphous formulation is not adequate because large polymers like dextran fail to protect proteins during drying. They stay nice and glassy, but they're too large to hydrogen bond effectively. And here's some examples of what this means in terms of protein structure. You can use an infrared spectroscopy to look at the protein secondary structure by observing spectra in the liquid and the dried state. So for this protein, interferon gamma, this is a second derivative IR spectrum. This dominant band here is due to alpha helix. If you dry the protein without any additives, you get this great perturbation of the spectrum indicating protein unfolding. And if you rehydrate that sample, you get this new band due to intermolecular beta sheet. This sample had about 18% aggregation after rehydration. In contrast, if you dry with sucrose, you retain a large degree of the native-like spectrum indicating protein unfolded is inhibited. You never get a spectrum that exactly matches the one in aqueous solution because in the dried solid, the loss of water itself impacts some of the spectral qualities. If you rehydrate this sample, you have a very tiny new band here for intermolecular beta sheet. This only had about 5% aggregation. Contrast what sucrose can do for structural retention to what happens with dextran. This is dextran and actin freeze dried. Here's the aqueous spectra with the dotted lines for actin. As you dry it with dextran, note that we get no indication of retention of native structure. So dextran makes a nice glass, but it's not effective at hydrogen bonding to the protein in place of the lost water molecules. Such interactions are critical for real-world storage stability of freeze-dried formulations. This is look at an interleukin-1 receptor antagonist. Turbidity after rehydration, which is an indicator of aggregation, goes up tremendously as a function of storage time if there's no sugar present. But as sucrose is included in the formulation, you can completely inhibit turbidity increases. Similarly, chemical degradation, in this case shown by cation exchange chromatography, which is indicative of deamidation, can be greatly inhibited as you have higher and higher sugar concentrations. So the glass transition temperatures of all these formulations were about the same, and the values were all above the storage temperature. The key factor here is the native molecule retention was better as there was more and more sucrose in the formulation. Here's another example showing the impact of both glass transition temperature and proper stabilizing additives. This is a interleukin-11 study. In the presence of the disaccharides, the native protein structure was greatly retained. In the presence of the polymer hydroxyethyl starch, there was no stabilization of the structure. But the starch gives a very high glass transition temperature. So just starch alone does not inhibit aggregation during storage. Disaccharides alone are fairly effective, but at 60 degrees, not great. And then if you have both the polymer to increase the glass transition and the disaccharide to stabilize the native structure, you get outstanding stability of this protein. Almost no aggregation after 9 months at 60 degrees storage of the dried formulation. So overall, when we think of compounds like sucrose, the mechanisms of stabilization can be through the Timoshev mechanism. If we have strong binding ligands, it can be through the Wyman linkage function. During freezing and freeze thawing, those same mechanisms apply, but during dehydration, we have the water replacement mechanism occurring. And during storage in the dried solid, the important factors are retaining the native protein and having a glassy matrix. So now let's go back to our two case studies. Case 1, remember, was reversible precipitation of the protein during freezing. And case 2 was irreversible aggregation due to freeze thawing. Excuse me. During case 1, if we added sucrose, it would make it worse actually, because sucrose increases the chemical potential of the protein, which means it reduces solubility. In case 1, the protein's irreversibly precipitating because freeze concentration is causing the protein to exceed the equilibrium solubility in the solution. Adding sucrose will reduce solubility and make the freezing-induced precipitation worse. Whereas in case 2, we have a aggregation occurring because the protein's perturbed by freezing and the unfolded molecules are coming together to aggregate. In this case, adding sucrose would help because it can actually effectively reduce the unfolding of the molecules in the subsequent aggregation. A bonus question to think about: what happens if we have a strong binding ligand? This actually helps in both cases. In case 1, a strong binding ligand increases chemical potential, which leads to an increase in solubility. Excuse me. Whereas in case 2, a strong binding ligand stabilizes the native state, which leads to less irreversible or less unfolding and irreversible aggregation. So knowing mechanistic insights into how things work can be very helpful to solving these practical problems. So now let's switch gears and talk about what happens at interfaces. Often if we do studies like agitation studies, this is data, these are images of the particles formed during agitation of a monoclonal antibody solution. You see these particles that look like bits of film that have been kind of crushed up. Excuse me. That's exactly what they are. If you imagine this soap bubble being an air bubble coated with the protein film, if you pop that bubble, you end up generating a lot of these particles. And the reason for this is proteins form films at interfaces. Here's a photograph of one of those films being drawn up from the air-water interface. Here's photographs of an air bubble with protein forming film and in chunks at the surface. And as you compress the bubble and expand it, you can knock those pieces off. So what happens is the film formation is the first step in interfacial induced aggregation or particle formation. That film can be disrupted by mechanical forces like compression and dilation of the air-water interface, bubble popping. Absorbed layers can be scraped off, and there may be multiple interfaces like air bubbles and silicone oil in a syringe. And any kind of mechanical force, including shock from dropping, can dislodge that film and particles. And here's a very nice early study that demonstrated this. This is looking at a monoclonal antibody at the air-water interface, and they did a simple thing. They just had a needle that would rotate, and each rotation it would rupture the air-water interface and break this film. Excuse me. And what they found is that rupturing led to a high level of light scattering due to particle formation, which could be inhibited by compounds like polysorbate, which are very effective at inhibiting the absorption of protein to interfaces. We've seen similar things with proteins at the silicone or oil-water interface, where the rupture of the interface leads to aggregation, and if you have surfactant, you can inhibit that. The difficulty we have is interfaces are everywhere in the life history of protein products, from manufacturing to shipping and storage to delivery to patients. And during manufacturing, before the last filling step, there is a 0.2 micron filtration, which is very good for removing particles. Unfortunately, the pumps that are used afterwards often generate more particles, and those particles will be delivered to patients unless a filter is used during administration. So oftentimes, if people have particle problems, such you see here, visible particles in a pre-filled syringe, the filling process is to blame. And this occurs because of numerous stresses. There's interfacial stress because proteins can absorb to the walls of the pump, either peristaltic tubing or the the walls of a piston in a cylinder and be scraped off or ruptured. Particles can shed from pumps material. There can be cavitation, particularly in rotary piston pumps and with valve action and time pressure filling. And importantly, for time pressure pumps, the pressurized solution, when it goes into the vial or syringe, will have outgassing of bubbles, which leads to more interfacial stress. Well, here's one example from peristaltic pump filling of a protein intravenous immunoglobulin. 3 different brands of tubing over a large volume of pumping with 10 ml aliquots taken. Microparticle levels vary depending on the tubing. This one brand of tubing, PharMed, results in the fewest microparticles. Nanoparticle levels vary also, with the PharMed tubing leading to the most nanoparticles. Interestingly, if you do a post-pumping stress such as agitation, the solution pumped through the PharMed tubing makes the most microparticles during post-pumping stress, and the thought is that's because nanoparticles that were formed during the pumping itself causes a lot more microparticles. And you can see that nanoparticle levels actually decrease during agitation, presumably by agglomerating together to make new microparticles. These results show mechanistically the roles of different particles formed during pumping, during post-pumping stress. They also point out that it's important to use proper methods to study protein particles, such as flow imaging and nanoparticle measurements. Measurements made by light obscuration would often miss these kind of effects because light obscuration is fairly insensitive to measuring protein particles. Okay, I wanted to give one last example, and this is trying to bring a lot of things together. And looking at the roles of conformational and colloidal stability on agitation-induced aggregation of a protein. And in this study, the protein was agitated and recovery of monomer was a measurement of aggregation. So as monomer levels go down, we're losing protein to aggregate. So at pH 7, there's a modest loss of protein to aggregates. At pH 3, this is almost completely inhibited during agitation. And at 5 and 6, you get a little bit more aggregation. If we correlate these results to measurements of colloidal stability, B22, what we see is at pH 7 we have a modestly positive value, meaning charge repulsion. At 3, the value is much more positive, and that charge repulsion is inhibiting aggregation during agitation. Whereas at pH 5 and 6 we're slightly negative or almost 0, which means there's very little repulsion, and during agitation, protein molecules readily come together. So for the pH effects, colloidal stability seems to play a role even during agitation. If we look here at formulation effects, at pH 7, if we add polysorbate, we can completely inhibit monomer loss due to aggregation because the protein is inhibited from adsorbing to the air-water interface, and it's really not anything to do with the intermolecular interaction energetics. If we add salt, we lose a lot more protein, and if you look, that's charge repulsion is lost in the presence of salt. We go to a negative value of B22, which means intermolecular interactions are favored. So charge shielding by the salt will promote aggregation. The really interesting results are what we get with sucrose. Sucrose, which is a really good conformational stabilizer, for this protein is a colloidal destabilizer. That is, sucrose favors intermolecular interactions, and the net effect of that practically is sucrose promotes protein aggregation during agitation. Even though it would stabilize the protein in solution, by favoring protein intermolecular interactions, sucrose destabilizes the protein formulation during agitation. So it's important to consider the actual stress against which you're stabilizing a protein. And in this case, obviously, polysorbate is the best stabilizer. Okay, let me conclude and say that we know in bulk solution that both conformational and colloidal stability are important for governing aggregation of proteins. And by stabilization with additives can depend on specific ligand binding through the Wyman leakage function, for example, or nonspecific stabilization through the Timoshev mechanism, This also applies during freezing. Whereas during drying, as we strip the hydration shell off the protein, water replacement is the mechanism. And finally, when we have interfaces, interfacial stress, particles are formed because films of the protein form, and any kind of disruption in that film will lead to particles in the bulk solution. And surfactants are quite helpful at inhibiting this, but obviously with modern research, we have to be aware that surfactants cause their own problems in particle formation due to degradation. And finally, read the literature. The newest literature is critical to keep up with, and the old literature back even decades ago has very important fundamental information. Well, I thank you very much for your attention. If you have any questions, my email is given at the beginning of this presentation. Please feel free to contact me. Take care.
Okay, thank you, Professor Carpenter, for that illuminating presentation. So it was a great presentation with a lot of mechanistic insights that we face in the real world. Briefly, we came to know about the reversible and irreversible precipitations, the role of specific binding, thermodynamic stability, Wyman linkage function, and how sucrose impacts the stability in these processes. Also, we came to know about the conformation and the colloidal stability and the freezing impacts on the which are also dependent on these stabilities. We came to know about the good buffers and the bad buffers during lyophilizations and how surfactants can impact them. We also came to know about how the interfaces and particles are a critical process in our manufacturing and drug development processes, and how the surfactants can impact them, although they have their inherent problems. So it requires a lot of understanding of each molecules. Even for people, us in biosimilars, I think having a formulation doesn't mean we are good to go. We have to evaluate each and every step for all these processes that has to be considered during a biosimilar development too. For this, I would like to say thank you once more and pass over the mic to the organizers, please.
Yamini madam, would you like to add something?
No, it was a very informative session. Secondly, because we are using such buffers in injectable formulations, we had in-depth knowledge from Professor John Carpenter about this. So I'm very thankful to him for his valuable insight into the protein aggregation mechanism and how to avoid protein aggregation and stabilization. Thank you, Professor John, and thank you, Dr. Pinaki Basu, for chairing the session.
Thank you, ma'am. Thanks.
Yeah.
So now I would like to request Dr. Pinakin Basu sir to please hand over the virtual certificate to Dr. Carpenter sir.
Hi, John. It's a great pleasure and honor to chair this session for you. And please accept this certificate as a token of appreciation for the pain that you've gone through in making this presentation available for all the bigger audience. I know it's very early up there, and it's probably you're chugging a lot of mug of coffees. But I really want to thank you for being in this presentation. Thank you very much from all of us here.
Oh, thank you, Pinaki, and thank you for the opportunity to participate via video presentation for the first part. Yeah, it's pretty early in Colorado on a Sunday morning, but I greatly appreciate this, and I'm really glad you guys put this conference together. I think it's extremely valuable for your colleagues. Thanks so much. Thanks for the certificate.
Thank you
Thank you,
very
John.
much, sir. Thank you. Now, I would like to request Professor Yamini ma'am to please hand over the virtual certificate to Dr. Pinakin Basu sir as a token of our love and gratitude.
Dr. Basu, please accept the certificate for chairing this valuable session from all of us. Thanks a lot.
Thank you very much. Thank you very much for the appreciation, and I'm glad to be a part of it. Thank you.
You're welcome. You're always welcome.
Thank you.
Thank you very much, everyone. So, madam, we'll be taking few minutes break now.
Okay. So we'll out of this admin session. We'll wait for a while. Then you tell us.
Now you are visible. So I think
Yes.
we need to help Dr. Carpenter because I think he's not able to hear.
No, he was the
So now we are taking 5 to 10 minutes break. Thank you, everyone.
Yeah.
आयुष, ओके, बट मैं बोल रहा हूँ सुनता है कि नहीं? टेस्ट हो सकता है? ओके
Sir, we can hear you.
ओके फाइन थैंक यू. So Divya ma'am, who else is there?
Sir, Yamini madam is there, you are there, and I am there, and we are waiting for some other members to join us, and then we'll start the valedictory function.
Sure. So is clear, right? Whatever I'm talking, or any echo
Yes,
is.
sir. No echo is there. You are clearly visible and audible.
Thank you so much.
Yes, sir. Hello, Dr. Savle sir, can you hear us? Hello.
I think he's not there.
Okay, sir.
Dr. Divya, if you want Dr. Savle, we can just, one of us can go out, then you can invite Dr. Savle because I think we are six.
Okay, I am going, madam.
Okay, okay. Dr. Divya, please invite Dr. Savle. I request one of the anchors to invite Dr. Savle and start the proceedings.
Yeah, good evening, ma'am. I'm in the room.
Yeah. No, I just request. I'm requesting the anchor to start.
Salam alaikum. So good evening to all. I think we are ready to go ahead with the valedictory function. So we have with us Dr. Ansuya Madam, and we have Dr. Savle also. Before we proceed for the valedictory function, I request Dr. Savle to announce the course. Over to you, Dr. Savle.
So what's our next thing?
We are going to show you a video. We are going to initiate one course on biotech. It's going to be a certificate course. So they are going to show a video in a short while.
Yeah.
Dr. Carpenter, please.
Okay.
Hi, Dr. Carpenter. Nice to have you in the room. Sir, I think Dr. Savle is facing some issues for his internet. Can we launch the video, sir? Sir, you are muted, I think.
We can start that.
Yeah.
That itself will be explained.
Yes, sir. Then I'll share the video. Yeah.
Please.
Yeah.
Welcome you, Dr. Carpenter.
Thank you. I'm going to step away for a second to get this bright sunshine off.
We at LM College of Pharmacy are excited to announce the launch of certificate course on biopharmaceuticals product lifecycle in collaboration with Intas Biopharmaceutical Limited. This course has been tailor-made to cater the need of understanding the life cycle of novel modalities of the therapy. Various experts with vast experience in the field of biopharmaceuticals will come forward to put this course together, which will have amalgamation of online learning and offline hands-on training. The course will help the students and aspiring graduates to develop skill in niche sector and to be industry ready.
Extremely pleased to announce the launch of a highly thought after certification course for biopharmaceuticals. Intas Pharmaceuticals and LM College of Pharmacy have joined hands to develop this curriculum, which will focus on the basic aspects of various current-day biopharmaceutical industry practices, and will also provide practical trainings to the students and interactions with industry professionals. This will become an essential course for students and industry professionals starting their careers in the biopharma industry. This will enable them to be industry ready and also provide them the flavor of various biopharmaceutical sectors currently operating in India. This is a much needed program, as this will provide students awareness in the field of biosimilars, novel biologics, vaccines, cell and gene therapy, and help them in stepping into the industry of their choice with great confidence. I'm proud to be part of the journey. I wish all of us very well. Thank you.
So we are
Details, just a minute. Details of all will be available on our website very soon.
Yeah, thank you, sir. So as you have seen in the video, like we are joining hand with Intas Biopharmaceutical Limited, and the course will have the elixir of gene therapy, cell therapy, biologics of maybe vaccine and the monoclonal antibodies, starting from cloning to upstream development, its purification, and lastly the formulation aspect. So the details will be available soon on our website. Moving towards the valedictory function, I request our beloved principal, Dr. Mahesh Shabaria, to address the valedictory notes. Sir, you are muted.
Thank you, Vivek. Dr. Ansuya Badalkar, Chief Guest of Honorable Dr. Carpenter, Guest of Honor of Therapeutic Function, Organizing Secretary, the invited guests, and dear participants. I'm sure for all the participants, this the evening of 4 days, last 4 days, is knowledge gaining and more insightful, specifically for the biologics. And there are many takeaways from this e-conference on biologics in healthcare. It's sure that the biologics, biopharmaceutics, gene therapy will be the future therapeutic age for many ailments. So to understand about that, know or to have the insight about this biologics was very essential. And keeping that thing in mind, we have organized this international e-conference for 4 days. We have received a support from Gujarat Biotechnological Business, and they are always supportive in organizing such events. And this support always motivates us to the best in the field wherever we are and whatever we are. I'm thankful to the GSPTM. We acknowledge the Thermo Fisher also being a industrial partner. During this e-conference, we have received around 1,450 registrations and around 13 eminent speakers across the globe have given their talk or they share their knowledge and experience with the participants. And the topics covered in this have covered the majority of the areas deal with this biologics. Some of the topics, the name, the basics of clone and cell line development for biosimilar projects, shift of biological research and future prospects, vaccine development for COVID-19, regulatory strategies for successful development of biologics development, mRNA-based therapy, gene therapy for hemophilia, immunogenicity of peptides and biologics. So these were the major topics covered in this particular e-conference. Participants were from majorly from India and 5 other countries also participated. The participants from industry, academia, and the research organizations also participated in this conference. We are specifically thankful to Dr. Carpenter for accepting our invitation to give a talk and be a guest of honor for this valedictory function. Thank you very much, Carpenter for accepting our invitation. Last but not the least, the compliments to Dr. Yamini ma'am and the team LLCP for organizing the wonderful e-conference. And all the speakers were really have given a very nice talk, very insightful and knowledgeable talks and discussions were there. So hopefully all the participants have enjoyed this knowledge enhancing 4 days. We'll come out with the many such e-conference in future also. Do join us again for the future program organized by the LFP. Once again, thank you all. Thank you very much.
Thank you very much, sir, for addressing the valedictory notes. Now I request our guest of honor, Professor John F. Carpenter, to share his few thoughts on biologics. Sir, over to you.
Oh, that's not much. I was really glad to hear about the course, by the way. I'm going to give you a little thoughts about education of students for working on biologics. Many of our experts in industry currently and the ones that will be in the future are not trained specifically on a lot of the aspects they need to know about for the development of protein-based drugs, vaccines, cell therapies, etc. Many of them come from very fundamental backgrounds, maybe biochemistry, and having courses that allow them to gain insight into specific industry issues is critically important. And anybody on the call that's in the Indian biotech industry, I strongly encourage you to support the universities for having these kind of courses, also to provide internship opportunities for students, and also to provide financial support to the universities directly, because the universities are training their future employees. And when they show up in your company, if they're not well trained in the specific things they need to know about for industry, you can help solve those problems for future leaders in your companies. So I just wanted to make that educational comment. I think it's critically important for the work we've done here in Colorado over the last 3 decades. Many of the projects were sponsored by industry. We've had outstanding collaborations with industry scientists, and we've had industry scientists mentor students. And those students go on, like Dr. Pinaki Basu, to be great scientists in the industry. Regarding biologics, it's more and more challenging these days because we have better and better analytical methods. Some of the products you're working on now to create biosimilars of have actually not the highest quality. You may have noticed if you analyze some of the innovator products that were created in the 1980s, 1990s, they have relatively high subvisible particle levels, for example. And we didn't know about that in the old days. We didn't have the appropriate instrumentation. And particularly if we only use light obscuration to evaluate particles in protein products, we would miss a lot of particles. Now with the modern techniques, we can actually understand what's going on better in the product. And I know you had a session on immunogenicity, and many biotech products, protein products, cause adverse immunogenicity in patients. For most of those products, we don't know the cause, but there are studies published and unpublished that directly show subvisible particles can be a contributing factor to immunogenicity. So subvisible particles can cause a miracle drug for a patient to fail in that patient. And I think we need to do a better job globally as industry to control and understand subvisible particles with the hopes that we have less immunogenicity. And again, because of biosimilars, with the new analytical methods, my hope is the biosimilars will actually be biobetters and can improve the quality compared to the innovator product. And the innovator products were all done as well as they could be at the time. They're good products, but many of them cause problems in patients. The other comment I wanted to make regards reading the literature, and this is for everyone. As I said in my talk, there's outstanding literature going back almost 100 years now, if not over 100 years, in critical areas like protein stabilization. And it's important to read that literature to understand fundamental mechanisms. But there's also really good literature daily coming out. And some of these papers you have to have library access to see, but many of them are open access. And let's take one modern problem, degradation of polysorbates. This is something that's been going on since the inception of biotechnology in the 1980s. But again, we didn't realize that it was happening in the field. And now there are many, many really good papers on how you recognize degradation of polysorbate, the particles they can contribute, and how polysorbates themselves can degrade proteins. And there are many good papers coming out on on the analytical approaches to use. So reading the literature is super important. And lastly, I wanted to make a comment about conferences. Hopefully next time this will be live. Everybody can get together in Mumbai or Delhi or somewhere for a nice conference and nice receptions and meals. I might even join you. My wife and my family and I have many friends and contacts in India. We've taken our whole family there for vacations. So we might come see you. But I think conferences are really important for the field. And just to mention again something about Colorado, we've had a conference on protein stability since 1994. And it's a relatively small conference, maybe 200 people, but it's gotten to be a very important gathering place, both for exchange of information and making new friends and seeing old friends. And I strongly encourage you to keep up the conference work, even if it's all online like this one, and to encourage participation from people from all aspects of the industry and universities. And then hopefully, as we get back together live, if you have a conference, obviously you want poster sessions. And really, one really important lesson we've learned over the years, make the poster session the only thing you do for part of a day, because the people that prepare posters deserve proper attention to their work. And actually at a poster, you can have the best conversations. And at our conference, we reserve a half day, and that's all we do is we have posters, and it's tremendously well attended. And that is encouraging for the students. It's encouraging for the postdocs and the scientists to know that their work is really well appreciated. And then one last comment. I'm an editor at the Journal of Pharmaceutical Sciences for reviews and commentaries. We get a lot of those papers from India. If you're an expert in an area and you know about some things you would like to tell the field about, either something that's fundamental and mechanistic or some really good insights from industry, please contact me regarding writing reviews and commentaries. And when you do so, let me know why you're an expert in the area, your papers you've published, your industrial experience. Those papers are really well regarded and actually highly read and cited. So that's it. Thank you very much. I really appreciate the opportunity to participate, and I think it's great people are sharing new knowledge and getting together like this at a critical conference to help educate everybody. Thank you.
Thank you very much, sir, for such an informative words with respect to biologics and how the polysorbate 80 is like a culprit since long for when it comes to formulations of biologics. We thank you again, and I request Dr. Divya to share the e-certificate, and I request Dr. Mahesh Chhabria to present the e-certificate to Dr. John F. Carpenter as a token of love and gratitude. Thank you very much, sir, again for joining with us and sharing your thoughts with respect to the protein stabilization.
Thank you for inviting me.
Our token of gratitude. Thank you for accepting our invitation.
Thank you.
So moving ahead, as we are near to the closure of the event, so we should have feedback, because we have gone through many different topics. We covered cell therapy, gene therapy, we covered upstream development, we covered downstream development. We also have a look at the end towards the formulation aspects of all this. We have also vaccine development. So entire biologics encompasses into the one event, that is Biologics in Healthcare 2021, organized by LM College of Pharmacy. So just we will have a look at a glimpses of the event through a feedback video.
Working in biosimilar formulation development, Conference Biologics Limited. It was a very great learning experience, very insightful sessions about the biosimilar formulation development, holistic approach for the process and product development, and regulatory aspects of this. Having had a chance to more new technologies and platform for the drug delivery development also. For that, I really would like to thank you organizers and sponsors for organizing such a nice events, and looking forward for attending this type of events and sessions in future. Thank you.
So thank you very much. Now, we move ahead, and now I would request Divya ma'am to please have the introductory slide of Dr. Ansuya ma'am. So it's my privilege to introduce Dr. Ansuya Adalkar. So ma'am is currently working as a Joint Director, Human Resource Development at Gujarat State Biotechnology Mission. Comes under the Department of Science and Technology. Before her current position, ma'am is a sector specialist for environment and marine biotechnology under the same Gujarat State Biotechnology Mission. She's had a biosafety cell, and she worked as a principal scientist in Marine Biosource Center, Jamnagar. So ma'am, I request you to please address the valedictory function.
I am audible?
Yes, ma'am.
Okay. So, good evening, everyone. All the members on the online dais and off the dais, all the participants and all the other members. Strangely enough, we cannot see anyone here. And so, but then yes, I believe there is a large crowd that is participating. I can see the numbers there. So it is very heartening to see that at this hour of the day as well, our participants are so alive and responding to the program. I have been through this program for all these 4 days because it was a very interesting content. So I could not resist the student inside me. So I was there all through till the end. And I must compliment that it was a wonderful program. This 4-day that we have spent hearing about biologics, it has been kind of a educational trip. So right from formulating the idea to having the product and all the intermediate chain that covers it. So I think it was an excellent program and an excellent initiative from LM College. I have got the opportunity to interact with LM College off and on through certain different other programs as well. And sir, I must say that you have a wonderful team. The last program that I attended, it was, I think, more organized by Dr. Mamta Shah. So the quality of programs always stay the same. So you hold on to that fort, like, you know, that you are the oldest pharmacy college, not only in the state, but also one of the oldest colleges in the country. So I congratulate you for that, sir. I would want to tell our participants that this LM College, it was formed by the initiatives of Ahmedabad Education Society. And this college represents a true mechanism that it demonstrates the mechanism of academia and industry linkage. Because this college was formed since we had a huge push from the pharma companies that we already had in our state. Gujarat has always been a pharma hub for very long, and that is how it very easily converted itself also into a biotech hub later. And LM College has played a very critical role in supplying the human resource that was very much required for the pharma industry. So this connect, I'm very sure it has always been there, and I hope it will always be there. And to a very pleasant surprise today, I got to hear about the certificate course. And it is very enthralling to know that, you know, students will get an opportunity here in our state to study biologics, how to manufacture it, and they can take their own initiative. We as JSBTM, we have one more program into entrepreneurship development. And there very recently, we have started seeing that we have participants who want to have their entrepreneur initiative into biologics. So I think it will be a very win-win situation. Sir, I will definitely come to you as well as you, ma'am, and get those participants to, like, you know, maybe participate in your program. That will really help them. I did not understand the kind of duration that the certificate course will take, but I understand it will be 10 months or something, or a year or so. So I think that will be very good for our participants as well. So a very encouraging initiative. Last 4 days that I have gone through this program, I also realized that this program we got to hear a lot of visionary speakers. Very startlingly, the words of one speaker is constantly resounding in my mind, and that is Professor Sarfaraz from the first day. When he talked about a very elegant example of Japan, he said that Japan never invested its time on science because they realized that the resources that they have, both monetary and human resources, are very limited. It's a very small country. So they invested all the time and all the energy in looking at technology. And today we know that, you know, the technology index, the patent index, it ranks very high. Nothing to beat USA, of course, but then yes, it is not very low on ranking. So this is a very inspiring word. As a government servant, I feel that these kind of inputs are very important for government reforms and policy reforms when we are looking at crowd as such, and whom to motivate and how to motivate and what to motivate in the people who want to do something good. So definitely listening to visionary speakers helps, and I am very sure through LM College we will be able to connect to more such visionaries in future, and that will be very nice. talking a little bit about the kind of changes that are happening in the pharma industry. I think I remember when at the time of my grandparents, the kind of diseases that were there, they never resounded in the time of my parents. So my parents faced a very different set of diseases, and today we as a generation, we are facing a very different set of disease. So this decadal map, if we study, and it is very strangely, it is not influenced by population. The population has been increasing at a very similar rate over last 3 decades. Maybe it has, like I was seeing some review somewhere. So the population has grown every decade by 20-21%, not more than that. But our disease map has changed drastically. In 1990s, we had diseases which were more enteric, which were more caused by maybe protozoans and some other elements which were like, more gross. Then came the 2000s, and we had the diseases that were like, we had never heard earlier. Enteric diseases did stay a little bit, but with it started all the respiratory disorders, the flus. And then we little bit had the genetic disorders. They just started showing up their head. And 2011 onwards, that we see, we have a whole capturing situation of genetic disorders and something that we would call lifestyle diseases. So these lifestyle diseases are something very strange, and addressing those diseases is also a very different ballgame. So scientists are endeavoring, I think, right from the 90s, from the time that they understood how biological sciences can help develop new drugs. The whole ballgame has kept on changing every singular decade. So today we are stuck with lifestyle diseases like deficiency diseases. Then there are some rare diseases like Alzheimer's, Parkinson's, and these numbers are now growing. So I think when we look at the past 3 decades and today, when we look at the disease map, I'm very sure that we can have a predictive disease map, like, what is going to come up next. And I think this college, being one of the pioneers and the foundation of pharma industry in our state, I'm very sure LM College will take up to this call, mapping diseases and also having a predictive science to see what diseases are upcoming. So when I talk of genetic disorders, I can see the way the map has changed. Earlier, the earliest study that I could trace to Gujarat, in Gujarat, the Verawal region was more affected by genetic disorders than rest of the state. Now it is spreading. So Kerala has become a very big hub of genetic disorders, and I think pollution and many other peripheral factors are causing these things. So emphasis on these peripheral areas is also very important. Also, as I can see a lot of potential in this college, sir, I will like to say that diagnostics is an area where, we have grown as a country, and as a state, I feel that we can do much more. We have much more potential, right from diagnostics, early diagnostics to prenatal diagnostics. These are going to be those areas where government will definitely want to have a push, have funding already ready for these areas. And these will be those fortes where colleges like yours can really become a hub and catering to not only to the industry but also making the human resource studying in your college aware of the potentials and the possibilities that they can do something. So we have a very good biotechnology policy which supports startups. So new entrepreneurs, if they have a very good idea, we are there to support them. So I think it can be a very good win-win collaboration for your participants of different programs to be associated with us for the policy benefits, because new endeavors are something that we really need today. Everything the larger pharma companies, I'm not very sure if they will be interested in, but there are smaller patches of problem areas where I think new entrepreneurs can do really well. Genetic diagnostics is definitely one such area. We are seeing that Bangalore has done real well in that area, and some very new entrepreneurs, very young entrepreneurs have come up and they have established their companies. They are doing fantastic. the samples from our state are going to Delhi and Bangalore for analysis, because today the urban population is awakened to this new need of prenatal diagnostics and genetic diagnostics. So since we have now a demand, I think it is very important we create a supply as well. And this new breed of entrepreneurs is something we are looking forward to. And I'm very sure your college will do something about it. As far as I can see, the Government of India has established a wonderful mission called Biopharma Mission. So this mission, I encourage all the participants who are here, as well as the rest of the young faculties who are associated with different colleges, to definitely go visit the website of National Biopharma Mission. All the archived calls that you will see there are primarily to do with biologics, biotherapeutics, diagnostics, genetic disorder diagnostics, and all these areas, which are very, very important. Central government is funding it heavily, and so is the state government. State government also gives a good amount of support for these areas. So I encourage all participants to look into these websites, so much so that DBT has also formed a national facility for biopharmaceuticals in Mumbai, which is a very good facility. It is very complete in itself, and any entrepreneur or any student who needs a training, they are there. Their mandate is to train. So these are some of the things that I want your college to take complete benefit of, because you are very well placed in this area and you are already ready with everything. The certificate course will do wonders to our state, no doubts in that. And any help needed, anything needed from GSBTM, we are always there. And last but nevertheless, Yamini madam, I will like to request you that apart from a longer certificate course, if you can also think of doing smaller skill development programs, smaller packages where, like, students can be trained into a very limited area of biologics, but hands-on. So, and we are also here to support, and that is all I want to share with the participants and the faculties here. And congratulations once again, and all the very best for all the endeavors. Thank you.
Thank you, ma'am. We'll take up your suggestions and we'll definitely do something in that area, like doing workshops, hands-on training, etc. Along with our industry partners, like we need their support as well, and we need finances as well.
Right. Your industry support is, I think, a very big backbone. And without them, it is difficult. So thank you very much for giving this opportunity.
Thank you very much, madam, for your inspirational and motivational words. Like, indeed, I agreed with you. Like, due to advancement in technology, like we are having everything very fast. So, like, due to application of machine learning and AI, like we are reducing the number of experiments from like 1500s to like almost 15. And we are happy to hear from Dr. Niazi that, like, after encoding the gene of COVID, the Moderna is ready with its first prototype within 3 days. So that is a huge success, and it's like first time ever that mRNA vaccines being applied to human beings. So that is very good technological advancement. Thank you very much, ma'am, for your motivational word. Now I request our beloved principal, Dr. Mahesh Chabaria, to convey the e-certificate as a token of love and gratitude to ma'am.
Ma'am, please accept our token of gratitude.
Thank you.
Thank you very much for accepting our invitation and for your support and encouragement. Thank you.
Thank you, ma'am. Thank you. Now I request Dr. Yamini Disha to have the vote of thanks and the concluding words for our Biologics in Healthcare 2021. Over to you, ma'am.
Yeah, it was a great event. Dr. Ansuya Lahiri Badalkar, our chief guest of the valedictory function and joint director, GSBTM, DST, Government of Gujarat, Professor John Carpenter from University of Colorado and University of California, our principal, Dr. M.T. Chabaria, all my LOC members, my faculty colleagues, all the participants all over India and across the world. It's been our pleasure to organize an international conference on biologics in Healthcare 2021, Unfolding Genes to Proteins, from 4 to 7 February 2021. Our conference has been sponsored by the Gujarat State Biotechnology Mission. Thank you, Dr. Ansuya ma'am, for gracing our valedictory function and sponsoring our conference. Our conference is also sponsored by our industry partner, Thermo Fisher Scientific India, for which we are grateful to them. Without our sponsors' generous financial support, it would not have been possible to organize such conferences. I'm also thankful to our guest of honor, Professor John Carpenter, for imparting such an in-depth knowledge and gracing today's function, valedictory function. I'm thankful to all our expert speakers who had immediately accepted our invite, shared their expertise in the field, namely Dr. Salunke, Professor Niazi, Dr. Maital, Dr. Ram Teke, Dr. Bakera, Dr. Lakshmikant, Dr. Ashok Mishra, Dr. Nitesh, Dr. Nidhi, Dr. Chatterjee, Dr. Rishabh, and Professor Carpenter. I'm also thankful to our members representing our national and international committee for their ready acceptance of our invite and helping us with their mentorship and support in the participation. I'm thankful to all chairpersons, namely Dr. Vivek Upasni, Dr. Damne, Dr. Trivedi, Mr. Rajiv, Dr. Shiv Prakash, Dr. Chavi, Dr. Vyasa, Professor Sarat, Dr. Ashwin, Dr. Udaya, Professor Dave, Mr. Nirbhay, and Dr. Basu for their valuable time and readiness in chairing the session, also immediately accepting our invite and gracing all our talks with their presence and expert comments. I'm thankful to our beloved principal, Professor M.T. Chabaria, Dr. A. Mehta, Dr. Anuradha Gajjar, Dr. J.B. Dave, Dr. Praful Baradia, Dr. Ambisha for their mentorship, continuous support and help. I take this opportunity to thank all committee members, namely scientific, finance, registration, technical, media, publicity, anchors, feedback, quiz, and attendance certificate, and attendee welcome committee. I'll just speak their names fast because I want to take their names, because without seeing day and night, they've been really helpful to me and supportive. Dr. Ketan, Dr. Radhika, Dr. Palla, Dr. Preeti, Dr. Sneha, Dr. Mittal, Miss Divya, Vivek, Dr. Jibisha, Dr. Mustaqim, Dr. Devang, Dr. Sandeep, Dr. Jayesh, Dr. Dipeng, Dr. Kunjan, Dr. Mehul, Mrs. Mittal, Mr. Nayan, Dr. Nayan, Dr. Vanita, Dr. Mukesh, Dr. Sandeep, Dr. Tejas, Dr. Jinali, Mrs. Krupa, Dr. Dharmishta, Dr. Chirag, Dr. Sunita, Dr. Savle, Miss Divya again, Mansi, Miss Mansi, Miss Karuna, Miss Mittal, Dr. Jayesh. I would like to thank everyone who has helped us directly or indirectly in organizing this event. They have been really, all people have been really helpful to me with their valuable comments in time, help towards the improvement of quality of the technical sessions and in general all the programs of this conference. All of them made our speakers, chairpersons, and our participants feel comfortable in carrying out their functions in time. I acknowledge their unwavering support in all the matters. I thank, last but not the least, I thank all the participants for their enthusiastic participation. My special thanks goes to all the my department colleague whom I have named, Dr. Vidhi, Dr. Tushar, and everyone whose name I have already spoken, because they have spent their precious time in organizing and coordinating inaugural valedictory and technical session. I'm thankful to Mr. Manish Jain for smooth operation of Webinar Jam platform. I wish you all safe and healthy future. Goodbye and good night. Thank you all.
Thank you very much, madam. Now we are towards the concluding of this event. So I request all to join us for the national anthem. Then we'll conclude the conference. Thank you very much. Just a minute. I think due to copyright issue, it is not allowing to play on YouTube. I'll start by another link.
जनगणमन अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड उत्कल बंगा विंध्य हिमाचल यमुना गंगा उच्चल जलधि तरंगा तव शुभ नामे जागे तव शुभ आशीष मांगे गाहे तव जय गाथा जन गण मंगल दायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे
Thank you very much. The link for the feedback is already being shared in the chat box, and you will also receive it in the Telegram group. So that's it from our side. We concluding the Biologics in Healthcare 2021. Maybe we'll see you in the next year, Biologics in Healthcare 2022. Thank you very much.
Thank you.